Hi everyone, welcome to another update from Parkinson's Queensland. Today I'm really excited to tell you that we're at the Queensland Brain Institute in one of the labs, and I'm here with Dr. Margreet Ritter. Margreet, welcome to the Parkinson's Queensland family.
Thank you for coming here to see us. Yeah.
Yep, really happy to be here. Margreet's a research fellow here at the Queensland Brain Institute. Margreet, perhaps you can share with our audience a little bit about your research work.
I'm, as he mentioned, a research fellow here at the Queensland Brain Institute, and I research the neural circuits involved in movement and related to Parkinson's disease. So we know that Parkinson's is a neurodegenerative disorder where some of the neurons and the pathways that are involved in neurons are affected. So what we're doing and what we can do behind this rig is we can record from specific neurons and see how other neurons influences their activity. And we've discovered a new pathway that's involved in motor initiation. And the idea is that if we can discover new pathways, we can kind of circumvent those that are affected in Parkinson's disease. So really enabling people that are struggling to initiate movements, which you can see with freezing of gait, to kind of get them moving again.
And of course, it is Science Week this week, so we thought we'd come and speak to Margreet to demystify a little bit about research, but also for all of you to understand some of the rigor and the processes behind research projects, particularly as they are related to Parkinson's disease, but also more generally. So Margreet, perhaps you can share again with us a little bit about the process in undertaking a research project and the rigour that goes behind it.
Absolutely. So I think it's very important that you think about the design before you actually start. So here actually we have a little video on the screen where we can record from a single neuron. So if we find that these particular neurons are involved in movement and we want to see how certain drugs interact with these neurons, we kind of, before we start, want to think of the effect size. So generally, if you expect a really big effect of a compound that you're testing, you need a smaller group size. So if the effect is like a traffic light turning from red to green, and that happens consistently after drug application, you'd only need a couple of cells to show that this specific compound has a big effect. But when the effects are more subtle, which happens in most types of researches, you want to really be confident that this subtle effect is not just happening by chance or, by fate. So in that case, you generally need bigger group sizes, and you also need control groups. So if you're saying that the effect is specific to a subset of neurons, you need to take a different subsets, and so that this effect is actually not happening in those other neurons. And then often you have what we call real control experiments where you don't apply the drug, but you just apply usually what the drug is dissolved in, which can be salt water, and show that if you do exactly the same experiment, but you leave out the actual drug, that you're not seeing the same effect. Because sometimes when effects are subtle, you can see changes in neuronal activity that are actually not due to the compound that you're testing. And then another thing that we really try and do here in the lab is do controls, what we call blind. So if you're testing two different compounds, I might have one of my colleagues label the tubes 1 and 2, so I don't know which tube actually contains the compound and which tube contains the control, so that when I'm here in the day performing the experiment and recording what is happening, I am basically blind to which cells got the drug and which cells didn't get the drug. Now this isn't always possible. Then what you can do when you have your data collected, you can analyze the data without knowing which data set belongs to which group. So I can have all my different recordings, and I'll get a colleague to rename the files, so they know in the end which files contain the groups of cells that got the drugs and which didn't. So that way you can basically know that your results are reproducible around the lab by other worlds, and that you're confident that the hypothesis that you've tested has a real clear outcome.
Now, obviously, that's in a lab environment, but perhaps you can share with everyone what's involved in taking something to human trials, because ultimately, that's where I guess a lot of people contact us to find out about different projects or different things that they may have tried.
Yes.
Perhaps you can just sort of again, simplify it and demystify it for people as to what it takes to get something to human trials.
Yeah, so I think the key point is safety. So before you can start testing a new drug in a large group of people, you want to make sure that it's safe. So I think there's different steps to human clinical trials, and one of the first steps is testing the drug in a small group, often healthy controls, just to see if it's safe. And then it goes to bigger groups, which usually involves a couple of thousand people, to see if the compound is actually effective, because that's the whole goal for the therapeutics, is can we cure or improve a certain disease. And then they also look at smaller side effects, so like headache or nausea, so it might still be beneficial, but you just want to make sure that you have everything covered and people know what to expect. And I think the big thing is it always gets done in large numbers. What I explained as well with our results, if we know that there are subtle effects, like 1 or 2 is just not going to give you the answer whether it's really effective. And I think everyone's heard about a placebo effect as well. Sometimes you can test something and it works on 1 or 2 people, but it's actually not really effective. And when it comes to neurodegenerative disorders, usually there's a range of disease progression. So some people might get really sick really quick, whereas other people get sick really slowly. So you need to be very careful when you say, well, this treatment helps, because it might just be that in this small group, these people would have had a much slower progression of their illness anyway.
Yeah. So, as you can imagine, Parkinson's Queensland being the peak body for Parkinson's disease in the state, we hear about people, trying their own homemade remedies. If you're at a barbecue, for example, and someone says, you hear someone saying, oh, look, I've got Parkinson's and I've tried this great homemade remedy and it works, you should try it. What would you say in that situation?
I think it's really hard because, people are, I think, responsible for their own health, and also, healthy lifestyle has really shown to benefit many diseases. So I think we all know from experience that if we drink too much alcohol, eat too much junk food, don't sleep enough, don't exercise, we're stressed because work's busy, family life's busy, that our bodies just aren't happy. And I think doing what you can to get yourself in a healthy lifestyle is not just beneficial for people with Parkinson's, but I think very beneficial for everyone. So if this person at the barbecue is saying, look, I've been drinking 2 cups of green tea and I just feel a lot better, if you happen to like drinking green tea, why not? But I don't think that those kind of lifestyle remedies should be a replacement for seeing a healthcare professional, because Parkinson's disease is a neurodegenerative disorder. Like, there's really stuff happening in the brain. And of course, I think COVID has shown us that there's a lot of misinformation going around, and there was a lot being advertised, even to a point where someone said that drinking bleach would kill COVID, and obviously we know that would not have a happy end if we started drinking bleach. So I think people really need to be aware that a lot of things can actually do more harm than good. And even if it can't do a lot of harm, you don't want to waste a lot of your money trying all these things.
Exactly.
And just emphasize what we talked about, that when we go to human clinical trials, there's a lot of money, a lot of time, a lot of researchers that have developed all these safety guards to make sure that what eventually gets on the market has gone through a lot of steps.
Yeah. So as you can see, there is and rightly so, a rigor and framework around these types of research projects. The team here at Queensland Brain Institute do a phenomenal effort. As we all know, not enough money is spent on research, particularly Parkinson's, but research generally in Australia is well underfunded, and that's something that needs to change. So thank you, Margreet, for agreeing to speak with us today. Hopefully for all of you, it's demystified the research process a little bit. As I said, it's Science Week this week, so let's give a huge shout out to all of the researchers around the country that are working on different treatments and ultimately the holy grail, a cure for Parkinson's. So Margreet, thank you for the work that you do.
You're very welcome.
Great to speak
Thanks
with you.
for coming to see us.
Pleasure. Thanks everyone, and we'll see you at the next one. Bye for now.